Welcome to Philly Tech Connect podcast, where Philly's tech minds meet. CEOs, developers, dreamers. This is your space. Each week we explore the journeys of tech innovators. Join us and be inspired.
Hey everyone, Dave here with another episode of the Philly Tech Connect podcast. Today I'm speaking with Dr. Eisinger, a cancer researcher for over 20 years. She has been published in high-impact journals like Molecular Cell Cancer Discovery, the Journal of Clinical Investigation, and before taking the helm as a full-time CEO of SyzOnc in July 2024, she held the Ann B. Young Endowed Assistant Professorship in Cancer Biology at UPenn School of Medicine. Her laboratory was funded by the National Cancer Institute, the Department of Defense, the state of Pennsylvania, the Steps to Cure Sarcoma organization, and she has been an independent investigator with her own lab for 10 years, but left academia specifically to lead this enterprise full-time. Dr. Karen, how are you doing?
I'm doing well. Thanks very much for having me.
Thank you. I love speaking with individuals in the medical field. It's an industry I don't know a ton about, but I'm happy to learn more. I did a little bit of, research a priori, and I'd love, in to the extent that layman's terms is applicable, if you could tell us a bit about solid tumors, the current treatments, and how you guys are approaching it.
Sure, no problem. Solid tumors are really the next frontier in terms of oncology. We've had a lot of success over the last 20 years with cell therapies and immunotherapies for tumors that are a little bit less complex in terms of their microenvironments, like leukemias and other kinds of what we call liquid tumors versus solid tumors. And what makes solid tumors a unique challenge is that they have very complicated infrastructure. So I often describe solid tumors as like apartment buildings. If you think of the residents of the apartment building as the cancer cells themselves, you can think of them as kind of a pretty minor component, actually, of the whole ecosystem, and it's also an ecosystem that has a lot of infrastructure. So with the apartment building metaphor, it'd be the walls, the plumbing, the electrical. Tumors have that type of complex, solid tumors have that kind of complex infrastructure as well. And the real source of failure for treatment strategies in solid tumor, in my opinion, over the last few decades, has been our inability to characterize that in the tumor infrastructure and the tumor ecosystem, and really come up with successful strategies that are going to help us resolve those complicated ecosystem issues that allow tumors to progress, to get worse, and to let the immune system in so that it can do its job. And so, really, the way that we focus on the solid tumor environment at SyzOnc is by taking into account not just the cancer cells themselves or just the immune system, as has been the strategy for really everyone over the last few decades, but we take into account all aspects, all critical aspects of the tumor ecosystem, that solid tumor infrastructure, the cancer cells themselves, the immune cells, the infrastructure that's made of what's widely called extracellular matrix, this dense protein structure that gives solid tumors their form and allows them to communicate with the immune system and with other cells in the environment, and really is a very powerful tool in the tumor's armory to avoid therapeutic intervention and to essentially evolve beyond any therapeutic approach that's been taken thus far. So we take all of that into consideration to come up with novel targets, novel strategies that will, for the first time, deconstruct or remodel that solid tumor environment and allow the tumor cells to receive anti-cancer therapy and the boosted immune response.
Fascinating. I used to live in an apartment building. I never thought of myself as part of a tumor cell, but now I
Very
will every time I go back. So solid tumors, are we talking like pancreatic cancer? Which are the ones? They're like the really bad ones that have like a really low survival rate, right?
That's 100% right. So we're talking about glioblastoma, which is a brain cancer. We're talking about liver cancer. We're talking about soft tissue sarcomas, which are tumors of connective tissue and muscle. Definitely pancreatic cancer. Um, and so, as you mentioned, these are tumors that are incredibly difficult to treat, that have been resistant to all the therapies that have been applied to them. And it's really, our view is that their ability to resist therapy is essentially they're living in this fortress of the solid tumor, which is heavily protected and allows them to essentially hide or evade all the interventions.
So you mentioned, well, at least in your bio, you'd worked at UPenn School of Medicine. It appeared to kind of be a well-funded, position. Now you're off on your own at SyzOnc, and I'm wondering sort of, why not leverage the resources that you had before? What sort of was the impetus for saying, oh, I'm going to go off and do my own thing, and this is why I believe I'll get a better outcome.
Yeah. It's a great question. I think in certain rare instances, it can be possible to um, commercialize technology from academia and be successful, perhaps maybe more in the medical device space or in other spaces. But when it comes to the development of therapeutics for, in oncology specifically, but in other diseases as well, the again, the infrastructure that you need really and the investment that you need to develop drugs and to develop a new platform for drug discovery really is not the academic environment really isn't built for that type of work. The academic environment is built to support much more of a basic or early translational work, and it's a lot harder to kind of recruit the kind of investment from the outside world that you need to get a drug to market. It can be in the hundreds of millions of dollars. And so doing that, spinning out companies it has been shown to be the most effective way of commercializing technologies. And so our company was originally a spin out from the Penn ecosystem. Spinning out essentially gives us the freedom to recruit investors, to be able to give equity in exchange for cash investments in the company, which is much more complicated when you have a university ownership or university licensing kind of monkey on your back. This is a much more there's a lot more freedom to operate in the startup space for us.
It makes perfect sense to me. I guess, and I wonder, you talk about needing maybe potentially hundreds of millions of dollars to pursue this. Maybe not all at once, maybe it's over time, but is there a chicken and egg problem here? I mean, on the one hand, you sort of like I would think you sort of want to be able to show some sort of promise, some promising trials, a reason why someone would want, to invest. On the other hand, without the funds to do so, how do you ever get to that stage? So how do you kind of approach it? What's kind of the process like?
Yeah, I think it's a great question, particularly for budding entrepreneurs in the biotech space. And I think that the system really does work very well in terms of someone with a technology they want to build or an idea. You develop it either as a student or a postdoctoral trainee or even as an independent investigator in the academic space. And that's what I did. I spent 20 years honing my knowledge, my experience, the work of my lab to identify what we most strongly believed was going to be a commercializable approach to novel cancer target identification and drug discovery before we spun it out. I think it's much more difficult to simply, graduate with your PhD and say, I'm gonna do drug discovery and change the world. What really has benefited us is 20 years of experience in the academic space, publishing papers, generating a reputation for our work, generating funding for our work, such that when we, go out and tell investors this is what they're doing and they learn about our team, they see that we're experienced, they see that we've been vetted essentially by the, NIH and by other cancer research foundations and the DoD for our studies. And that gives them faith and confidence that we know what we're doing and that they can invest with us successfully. I think it's much harder to get a company started without at least some team members that have that kind of experience coming out of the academic space. So it's certainly the academic space is certainly an essential part of the research and biotechnology ecosystem. But I think there comes a point, there's a precipice where, at least for cancer biologists, where you ask yourself, am I going to stay in the academic space and continue to publish papers and train the next generation, which is, what the vast majority of academics do? Or have I identified something from my own research that's so valuable, that has such potential, that I feel obligated to go out into the commercial marketplace to try to bring it to patients to get them better outcomes. And I think, I'm in an incredibly lucky position to have found that thing that I think is so valuable, is so commercializable, that is going to change the marketplace and revolutionize the solid tumor space entirely. And I remember, I was speaking to a few other academics at a conference several months back. And I asked them, and they're very senior people, and I asked them if they had ever considered starting a company, and they said, I never found my thing. I never found the thing that I thought could really make a difference. And I think that's where I feel really blessed and lucky, is I did find the thing. I did, we worked really hard to identify the thing that we thought was going to make the difference, and that's our platform for target identification and drug discovery. And then you as a cancer biologist, your whole being is about improving patient outcomes. And so at that point, you say, I have to take the path that's going to lead to the best patient outcomes, and then I have to decide for myself, is that path an academic path, or is that path in starting up the company and commercializing this idea? And for me, it was a very clear trajectory that the best way to improve patient outcomes with our work was to translate it through the startup, through the biotech ecosystem, into clinical tools for patients.
Well, I certainly wish you guys the best, because it would be great to see progress in a space that, like you said, sounds like it's stalled, relative to other sort of oncology endeavors. It looks like you've got the team kind of assembled, or to a degree, just from having looked at the website. What's next for you? Are you mainly kind of in the pitching phase and looking to kind of have conversations with investors, or is there something else going on?
Uh, yeah, I think, everyone realizes it's a really tough time out there for biotechs, for academics as well. All of science is struggling under, the new administration's priorities and issues around just the general market. For us, what we're doing is staying laser focused on our scientific progress for developing drugs. We have drugs that hit compounds that we're developing, which we hope will be drugs for patients someday very soon. And so we're staying very focused on that. And at the same time, of course, we're constantly talking to investors, constantly building relationships, which is, what investing is all about, is getting to know the various institutional partners and strategic partners that do invest in the biotech and oncology spaces, building long-term relationships with them so that as our fundraising needs grow, because this is an expensive process, we have those relationships in place to fund the work that we're doing. And we've been very fortunate. We just received notice that we've been awarded a small business grant from the National Cancer Institute. We were successfully able to raise a pre-seed round, even in really difficult times. And so, what we're doing now is focusing on meeting the milestones that are critical to our endeavor, which is, of course, developing our small molecules and identifying targets for additional tumors.
Like I said, fascinating. For individuals who want to learn more about you and your work, where should they go?
We have a website. It's SyzOnc.com. And you can send us an email through the website portal. You can also reach me at karen.eisinger@syzonc.com. We're always happy to engage with interested scientists, investors, and patients and families who are dealing with solid tumors. Their input on their experiences and what they'd like to see going forward is critical to us. It inspires us. There's nothing more inspiring for a cancer researcher than spending the day with, the community of cancer patients and patient families. They're why we get out of bed in the morning. That's why we do this incredibly difficult job. There's no greater reward than thinking we can make these people's lives better, we can make these people's lives longer, and we can take some fear out of their lives. That's for any cancer researcher, that is the pot of gold at the end of our rainbow. That's what motivates all of us to get the job done. And so we're happy to talk to everybody about what we're doing and what we think is necessary for success in the solid tumor space. And we're so excited about next steps and making progress. And we're happy to engage.
Thank you so much.
That's all for today's episode of Philly Tech Connect. Join our community at phillytechentrepreneurs.com. Special thanks to our sponsor, Shortlist.io, a digital marketing agency specializing in top-notch link building, content strategy, and web design. Stay innovative, Philly, and see you next time on Philly Tech Connect.